Dear students, now we are going to discuss the mechanism of photosynthesis. So the mechanism of photosynthesis is with with the help of two headings. One is a light reaction and second one is a dark reaction. So first pathway, first event is a light reaction. Mechanism of photosynthesis. Okay, so we divided a mechanism of photosynthesis into two parts: light reaction and a dark reaction. So first one is a light reaction. Okay, so first of all, we should know what about the role of a physical factor, that is light, in light reaction. So definitely the ultimate source of energy in this ecosystem is sunlight. So during light reaction, radiant energy, sunlight, means radiation, sunlight. Radiant energy is converted into chemical energy. What is that chemical energy? ATP and NADP, HH plus, reduced form of a coenzyme. So this is ATP and NADPH plus. These two collectively called as reducing power, as they are able to reduce the C of CO2 into carbohydrate, reducing power. Or assimilatory power. Assimilatory power, as they are concerned with the assimilation of carbohydrate from CO2. So here, which molecule is able to convert light energy into chemical energy? Definitely chlorophyll A molecule. Rest of the molecules, carotenoids, means xanthophylls, carotenes, chlorophyll B molecules are not able to convert radiant energy into chemical energy. So chlorophyll A molecule present in reaction center. Before this, we completed a photosystem 1 and photosystem 2. In each photosystem, one reaction center is present, that is represented by chlorophyll A molecule only. So only chlorophyll A molecule present in reaction center is able to transform radiant energy into chemical energy. Rest of the pigments are, they are able to absorb, they are able to transfer that energy to reaction center. Okay, so this conversion of radiant energy into chemical energy is achieved by, so such a transformation, synthesis of chemical energy or conversion of radiant energy into chemical energy, it is by so-called one. So radiant energy to chemical energy is by ETS, electron transport system, ETS. Simply I'm writing here ETS, electron transport system, electron transport system, ETS, electron transport system. Okay, so by the transportation of electrons, the radiant energy is converted into ATP and NADPH plus, hence the name electron transport system. Okay, so What about the electron transport system and how many kinds are there? Okay. So for electron transport system takes place by two methods. One is non-cyclic electron transport system. Non-cyclic electron transport system. Okay, and second one, cyclic electron transport system. So these two are concerned with the synthesis of or conversion of sunlight into chemical energy. So conversion of radiant energy into chemical energy takes place by means of both non-cyclic and cyclic. Okay, right? Now, what about a non-cyclic electron transport system? First one, non-cyclic electron transport system. First, what are the requirements? Requirements for a non-cyclic electron transport system. So first electron transport system is non-cyclic electron transport system, that is actually non-cyclic photophosphorylation. We will see what is a photophosphorylation. Synthesis of ATP in the presence of sunlight. Synthesis of ATP in the presence of sunlight. So non-cyclic electron transport system accounts for synthesis of ATP by light. Hence the name non-cyclic photophosphorylation. We'll see at the end, don't worry. What is that photophosphorylation? We'll see at the end or during course of completion of that mechanism. Okay, right? So here, what are the requirements? Okay, so in the case of higher plants, particularly in higher plants, one, granum thylakoids. In the case of eukaryotes, you can take eukaryotes. Okay, granum thylakoids. Inside the granum thylakoids, what are the electron carriers required? And what are the chemicals required? For non-cyclic electron transport system, first one, water is required. H2O or H2S. H2S in the case of prokaryotes. So use H2O. If you go back to Cornelius van Nile, instead of H2O, H2S was the hydrogen donor. Okay, right? Granum thylakoids. And you can take a chromatophores of blue-green algae also. Chromatophores of blue-green algae also. They are having non-cyclic electron transport system. Okay, so this is present in particularly higher plants, eukaryotes, and some prokaryotes with oxygenic photosynthesis. And bacteria, they are also with a non-cyclic electron transport system, but without H2O molecule. So this H2O is usually means oxygenic photosynthesis. So water molecule. Next, what are the photosystems required? Both PS1, photosystem 1, photosystem 2, both are required. Both must be operated. Next, pheophytin. Pheophytin. Pheophytin is a type of chlorophyll A molecule. This is one type of a modified chlorophyll A molecule. One type of chlorophyll molecule. One type of a chlorophyll. Chlorophyll A only actually. But in that chlorophyll A molecule, magnesium atom is absent. Okay, remember pheophytin. Moreover, it was not given in NCERT. Okay, additional statement form. Pheophytin. Next, plastoquinone. Plastoquinone. PQ. Next, cytochromes. Cytochromes. Cytochrome B by F complex. Cytochrome B and F type. Next, plastocyanin. Plastocyanin, a mobile electron carrier. It is a mobile electron carrier. Just here I'm giving components only. You have to remember all these components. Mobile electron carrier. And it is a copper containing protein also. Copper containing protein. Cu containing. Copper containing protein. And again, I will explain you all these components at the time of mechanism. Plastocyanin. Next, pyridoxine. Pyridoxine. Next, NADP plus. Next, ATP synthase complex. Okay, so these are the basic requirements to understand the mechanism of a light reaction electron transport system by, that is a non-cyclic electron transport system, non-cyclic photophosphorylation. Okay, so what are the key areas of a non-cyclic photophosphor? Important areas, main areas, important areas. One, photolysis or oxidation, photolysis of water or oxidation of water in the presence of light. Oxidation of water in the presence of light, splitting of water. Next, second one, creation of H plus proton gradient. H plus gradient. Third one, synthesis of both ATP and NADP H plus. Next, fourth one, O2 is evolved, oxygen. O2 evolved. Next, fifth important one, key points. Red light with two types of wavelength required. One is 680 and 700. 680 shorter wavelength of red light. 700 longer wavelength of red light. So in the presence of these two wavelengths of red light only, non-cyclic photophosphorylation is possible. Okay, so what are the main important areas? Photolysis of water, creation of a proton concentration gradient. Creation of H plus gradient means concentration gradient. Synthesis of ATP and NADPHH plus. Evolution of O2. Light required, red light. Quality of light, red. And what about the wavelength of light? Intensity of light. 680 nanometers, 700 nanometers. So here, during non-cyclic electron transport system, two types of photochemical reactions are taking place. One is run by shorter wavelength of red light. The other one is run by longer wavelength of red light, 680 and 700. So these are the requirements of a non-cyclic electron transport system. That accounts for synthesis of ATP. Photophosphorylation. Photophosphorylation means synthesis of ATP in the presence of light from ADP and inorganic phosphate. We will see during the process. Now, what about the mechanism of non-cyclic electron transport system? Okay, so now mechanism of non-cyclic photosphorylation. Mechanism of non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Okay, so this one is a okay, so this is a transverse section of a thylakoid membrane, granum thylakoid. So this is membrane, granum thylakoid. So granum thylakoid. Granum thylakoid. Granum thylakoid. Towards outer side, this is stroma side. Stroma. Stroma side. This one, lumen. Lumen side. Lumen part. Okay, granum thylakoid, lipoproteinaceous membrane with pigments. Okay, so now non-cyclic electron transport system starts with excitation of electron from a reaction center. What is that reaction center? So here reaction center is a P680. Okay, so P680 means first complex involved. One is photosystem, two involved. So 680 is the reaction center of a photosystem 2. Okay, so non-cyclic electron transport system is initiated by absorption of, for example, this one is a LHC, for example, only light harvesting complex, antenna chlorophyll complex, and sunlight. So radiant energy, particularly 680 nanometers of wavelength of light is absorbed by LHC. From there, it is transferred to reaction center. Transferred to reaction center. Here, chlorophyll A molecule is present. What is 680? 680 is nothing but a red light with a shorter wavelength absorbed by chlorophyll A molecule. So, chlorophyll A molecule present in reaction center source, highest absorption, peak absorption at a red light with 680 nanometers. So, in the atomic nucleus of chlorophyll A, so here chlorophyll A is present. Okay. So now I'm erasing this one. Chlorophyll A. Actually, this is chlorophyll A molecule. Chlorophyll A molecule whose peak absorption is at maximum absorption is at 680 nanometers. So once the energy levels of electrons is increased according to chemistry, what about the electron ejected from atomic nucleus? So before absorbing radiant energy, 680 is at ground state only. Ground state only, less energy levels. So now some amount of energy is absorbed by 680. That is electrons, the energy levels of electrons present in the chlorophyll A molecule is rising. So now electrons are ejected from atomic nucleus of chlorophyll A molecule. So now excitation of chlorophyll A molecule. So excited. So chlorophyll A excited. So excited one is star. Star like a symbol, the sign of that one. Excited. So excited one is able to donate electrons. Okay, so 2 electrons donated. Now chlorophyll A with positive charge means electrons lost. Now it is getting positive charge. So how many electrons? For example, here I'm taking 2 electrons. For example, only 2 electrons. Okay, so once the energy levels of electrons is increased, what about the electron ejected from the atomic nucleus of chlorophyll A molecule? So what is the role of light to increase energy levels of electrons present in chlorophyll A molecule? So once the energy levels of electrons is raised, what about that electron ejected from its atomic nucleus, that is chlorophyll A molecule? So now 2 electrons donated by chlorophyll A molecule. So chlorophyll A is oxidized or not? Oxidation, loss of electron. So now chlorophyll A is at high energy level, transition state. So now it is in oxidized condition. Electrons donated, loss of electrons, definitely oxidation only. That now it is in oxidized condition. So now 2 electrons accepted by first electron acceptor. Here first electron donor is a chlorophyll A molecule present in reaction center. First electron donor is chlorophyll A molecule present in reaction center of PS2. Now, first electron acceptor, pheophytin. Pheophytin is a first electron acceptor. So what about the location of pheophytin within the membrane? Towards stroma. Towards outer side or not? If you split this membrane thylakoid into 2 parts, make it half like this, upper part, down part. Towards stroma, towards lumen. This part is towards lumen. This one towards stroma, means outer side. So first electron acceptor is located towards outer side, means towards stroma, but within the membrane only. So now 2 electrons are accepted by pheophytin. Now it is reduced. After accepting electrons, reduced or not? So now 2 electrons are in pheophytin. What is pheophytin? One type of chlorophyll A molecule in which magnesium atom is absent. Magnesium atom is absent. So this is a chlorophyll A molecule. Now it is in reduced condition. Now come back. So chlorophyll A is deficient in how many electrons? 2 electrons. Who is able to compensate that two electrons to a certain number, whatever the number? Here I'm taking 2. So to come back to ground state, compulsory chlorophyll A molecule loss of electrons must be compensated, replaced, or should be given to that chlorophyll A molecule or not. Now towards lumen, in association with that complex, in association with PS2, one more complex is present. What is that complex? This complex is oxygen evolving complex. Oxygen evolving complex. Complex. Simply OEC, oxygen evolving complex. So within this complex, manganese. Go back to mineral nutrition. What are the inorganic nutrients concerned with photolysis of water? Manganese. Chlorine. Calcium also correct, but according to NCERT textbook, manganese and chlorine. Calcium also correct, but it was not given. Okay, right? So this complex is concerned with oxidation of, this complex is concerned with oxidation of water. So H2O gives rise 2 electrons, 2 H plus protons plus half O2, half O2, one oxygen atom. So this complex is able to cause splitting of water or oxidation of water. So according to statistics or requirement of photons, to cause oxidation of one H2O, how many photons are required? So photon requirement, photon is energy particle. So 4 photons, PHOTONS, photon energy particle, required for photolysis of one H2O molecule. 4 photons are able to cause photolysis of one H2O molecule. Remember that one. Okay. So what about the role of this complex concerned with the photolysis of water, splitting of water, or oxidation of water? So now, how many electrons are available here? 2 electrons are available. Now, these 2 electrons, with the help of a OEC complex, moving into chlorophyll A molecule present here. So now excited chlorophyll A molecule, after accepting 2 electrons, reduced and coming back to ground state to run the action again and again. So after accepting 2 electrons, now it is coming back to ground state. Okay, so who is the donor of electrons to chlorophyll A molecule? H2O. In textbook, one question was there. In oxygenic photosynthesis that involves 6CO2 plus 12 H2O molecules gives rise 1 glucose molecule plus 6 H2O plus 6 O2. That the question is, what about the importance of role of 12 H2O molecules? 12 H2O molecules are photolysed during non-cyclic electron transport system. If one H2O molecule is photolysed, what about the number of atoms produced, oxygen atoms? 1. So to produce 6 O2 molecules, how many O2 atoms required? Definitely 12. 12 divided by 2, 6 O2 molecules. So to produce 6 O2 molecules or 12 half O2 molecules, what about the number of H2O molecules required? 12 H2O molecules. Remember that one. So during non-cyclic electron transport system, according to photosynthetic equation that accounts for synthesis of one glucose molecule, what about the number of H2O molecules must be oxidized here? 12 molecules. Okay, just leave it. I'll come back to this one again. First, take a 1H2O molecule as the standard one to understand the mechanism. After that, we will take a complete non-cyclic electron transport system that accounts for photolysis of 12 H2O molecules. We'll see that one later after completion of basic mechanism. So now, first electron acceptor is towards outer side. Within the thylakoid only towards outer side of the membrane. Now, 2 electrons present in pheophytin accepted by next electron acceptor, that is a PQ, plastoquinone, PQ I'm using here, PQ. So PQ is towards down or not? Towards down. So what is a PQ? Plastoquinone. According to textbook, PQ is not an electron carrier, but proton transporter. It was clearly given, follow that statement only. So plastoquinone is not an electron carrier, but H plus carrier. It was given there. But according to my knowledge, plastoquinone is able to transport both electrons and protons. Actually, it is concerned with transportation of electron and translocation of protons. But textbook statement, PQ, so pheophytin, first electron acceptor is giving electrons, certain number, whatever the number, to not an electron carrier, but H plus transporting one. It was given like that. But anyhow, now, so what about the total number of electrons available? 2 electrons. So take 2 electrons. Here I'm taking 2 electrons at a time. So now 2 electrons are accepted by PQ. So now this molecule is taking up 2H plus from stroma. So now PQ is reduced. PQ is reduced into PQH2 and immediately oxidized. Immediately oxidized. So now this H plus carrier donating electron to next electron acceptor. So which one is the next electron acceptor? That is cytochrome complex. Here I'm taking cytochrome complex. This is cytochrome complex. Cytochrome B, C. C is nothing but cytochrome. Cytochrome B, cytochrome F. Cytochrome B F6, B by F6, B by F6. That is one type of cytochrome. So now electrons accepted by cytochrome complex. So while transporting 2 electrons, what about the protons? 2 electrons given to this one. Now protons, how many? 2 added to lumen. 2 electrons, 2H plus, 1 is to 1 ratio. 2 electrons to H plus, 1 is to 1 or not. So while transporting 1 electron from PQH2 to cytochrome complex, how many protons are released into lumen? 1. 1 electron, 1 H plus. 1 electron, 1 H plus. So 1 is to 1. Okay, right? So what is the role of the association of these 2 electron carriers? Translocation of protons from stroma to lumen. So far, what about the total number of H plus accumulated? 2 H plus coming from water molecule, and 2H plus withdrawn from the stroma. So while transporting one electron to this one, how many H plus added to lumen? 1. Okay, so this is called as a quinone cycle, quinone cycle, quinone cycle. One electron, one H plus, one quinone cycle. Second electron, second H plus. So here, quinone cycle, cyclic moment, cycle. PQ, PKH2, oxidized, reduced, oxidized, reduced, oxidized. Okay, so 2H plus removed from stroma and added to lumen part. Okay, through cytochrome complex. Next electron acceptor is a plastocyanin, PC, plastocyanin, copper containing protein, electron carrier, mobile electron carrier between cytochrome complex and the next one. We'll see what is that next one. So now PC is with 2 electrons according to number of electrons available from H2O molecule. Okay, right? So now PC is in reduced condition. After accepting 2 electrons, this one is oxidized and this one is reduced. Okay, so to accept 2 electrons present in PC, next electron acceptor must be deficient in 2 electrons. What is that next electron acceptor? Next electron acceptor is P700. That is a photosystem 1. First involved is photosystem 2. Second involved is a photosystem 1. Okay, so the nomenclature of photosystem 1 and 2, not on the basis of a sequence of involvement during non-cyclic electron transport system. It was on the basis of a time of or order of their discovery. First discovered one, 700. Next discovered one, this one. So before accepting 2 electrons from PC, 700 must be deficient in 2 electrons. So now LHC, light harvesting complex, red light with 700 nanometers absorbed by LSC, transferred to reaction center. Now excitation of 700, excited and donated also electrons, 2 electrons. So 2 electrons ejected from 700. So 700 is deficient in 2 electrons or not? Now 700 is able to accept electrons coming from PC, plastocyanin. So oxidation of PC depends on oxidized condition of 700. If 700 is deficient in 2 electrons, it is able to take up 2 electrons. So before causing oxidation of plastocyanin, 700 must be at excited condition and deficient in certain number of electrons, and that must be in oxidized condition. Okay, right? Next, these 2 electrons are accepted by ferredoxin. Ferredoxin is a copper containing protein. Go back to mineral nutrition. We'll get what is cytochrome and ferredoxin. So now 2 electrons are accepted by ferredoxin, reduced, and immediately undergoing oxidation. So now ferredoxin donating electrons to next electron acceptor. What is that next electron acceptor? Make it down. Okay, next electron acceptor is NADP plus. That is coenzyme. NADP plus present in stroma. Here enzyme required is a FNR. What is FNR? FNR is ferredoxin. F is the ferredoxin. NADP. R, reductase. NADP reductase. Hence the name FNR. Or simply This enzyme is also called as NADP reductase. NADP reductase. Okay, so that NADP reductase requires the presence of NADP. What is that NADP? Coenzyme. NADP is coenzyme. Go back to cofactors. Okay, right? So how many electrons here? 2 electrons. For the reduction of NADP into NADPH plus, how many protons required? 2H plus coming from stroma. 2H plus from the stroma. The source of protons is from stroma only. Don't use these protons. Okay, so 2 electrons plus 2H plus. Now, NADP plus is reduced into NADPH plus. How many molecules? Only 1. So electron transport system completed. So here, who is the ultimate donor of electrons? H2O. Ultimate electron acceptor is NADP plus is final acceptor of electrons. What is the role of light in both the cases? To drive electrons present in reaction centers of 680 concerned and 700 concerned. So these electrons coming from this one, see, PS2 is giving 2 electrons to 700. 700 giving 2 electrons to NADP. And ultimately, who is able to compensate electrons? H2O. Okay. So H2 is oxidized. Only oxidation here. H2 is undergoing oxidation only. Next, what about chlorophyll A? First oxidized, next reduced. Pheophytin, first reduced, next oxidized. Next, what about PQ? First reduced, next oxidized. Cytochrome complex, reduction, oxidation. Remember reduction and oxidation, redox reactions. So plastocyan, first reduced, next oxidized. 700, first oxidized, next reduced. Ferridoxin, first reduced, next oxidized. NADP plus only reduced into NADPH plus. So what is the role of this electron transport system? Driving electrons present in H2O molecule to the acceptor, that is NADP plus. So the role of light is to drive electrons present in H2O to acceptor, that is NADP plus. Electron transport system completed. So out of assimilatory power or reducing power, so far only NADPH plus produced. Now what is the ratio between H2O molecules and NADPH plus? Only 1:1 ratio. So out of photolysis of one H2O molecule, what about the number of NADPH plus produced? Only one molecule of NADP is reduced into one molecule of NADPH plus. Reason, out of one H2O molecule, what about the number of electrons available? 2 electrons only. So 2 electrons along with 2H plus coming from stroma are able to reduce one molecule of NADP into one molecule of NADPH plus. Here ratio is very, very important. One H2O molecule, one NADPH plus only. And one more thing. So far, within the lumen, what about the total number of H plus available? 2H plus, 2H plus. Remember this one. So how many H plus available? 4H plus available. 4 protons available. Remember that one. Now come back. What about again recap of all these things? So non-cyclic electron transport system is initiated by red light with 680 nanometers. So red light with shorter wavelength is able to initiate non-cyclic electron transport system by increasing energy levels of electrons present in chlorophyll A, that is reaction center of photosystem 2. Okay, so non-cyclic electron transport system is initiated by What type of photosystem? That is a PS2, by observing what type of wavelength of light? Red light with shorter wavelength of red light, that is 680 nanometers. So excited chlorophyll A molecule donating 2 electrons. So donating electrons means chlorophyll A is undergoing oxidation or not? Donor of 2 electrons. So who is the first donor here? Chlorophyll A molecule. And who is the acceptor of first electron acceptor? Pheophytin. Okay, next. How many electrons are deficient in chlorophyll A molecule of PS2 present in reaction center? 2 electrons, for example. Who is compensator of 2 electrons? H2O. What is this complex? Oxygen evolving complex associated with PS1 towards lumen. Lumen side only, attached to lumen side of the membrane. Outside the membrane, attached to membrane, facing the lumen. So this complex is concerned with the photolysis of water. One H2O molecule, oxidation, 2 electrons, 2H plus, half O2, 2 electrons with the help of OEC reaching the reaction center. That is a atomic nucleus of a chlorophyll A molecule that is coming back to ground state to run the reaction, same reaction again and again. Now, what about pheophytin? Donating 2 electrons to PQ. PQ is accepting 2H plus from stroma. So 2 electrons, 2H plus, 1:1 ratio. Now it is transporting that electron to cytochrome complex. While transferring that electron to cytochrome complex, 1H plus is added to lumen. Like that, 2 electrons, 2H plus, 1:1 ratio. First reduced, next oxidized, reduced, oxidized, reduced, oxidized. So while transporting 1 electron, how many protons are released into lumen? 1. 1:1 ratio. Now 2 electrons are accepted by plastocyanin, 2 electrons. So plastocyanin is reduced. Plastocyanin is a copper containing protein. It is a mobile electron carrier, that is towards lumen side on the surface, inner surface facing the lumen, like this one. But this one is a mobile electron carrier between this complex and this one, cytochrome and 700. Mobile electron carrier between cytochrome complex and 700. So 700 is able to accept 2 electrons coming from PC if it is deficient in 2 electrons. So before causing oxidation of a so-called PC, what about the reaction? Now LHC of a photosystem 1 absorbing 700 nanometers red light transferred to reaction center. Ejection of 2 electrons. So 700 is deficient in 2 electrons. Now it is able to accept 2 electrons coming from plastocyanin. So these electrons are passing through ferredoxin, finally NADP. For the reduction of 1 molecule of NADP, how many electrons required? 2 coming from H2 molecule. How many H plus required? 2 coming from stroma. Reduced into 1 molecule of NADP HH plus with the help of an enzyme, that is NADP oxidoreductase. NADP oxidoreductase is a conjugate enzyme, holoenzyme. That enzyme is concerned with oxidation of ferredoxin and reduction of NADP. Here coenzyme required for that oxidoreductase is a NADP is the cofactor, coenzyme. Okay, so oxidation of this one and reduction of this one. Okay, so what type of enzyme this one? Belongs to which class? Oxidoreductases. Concerned with oxidation of this one, reduction of this one. What type of cofactor is required? Coenzyme is required, NADP plus. So far, how many enzymes involved? So far, 1 enzyme involved. Okay, so it was not completed. Okay, so far we completed flow of electrons only, right? Is it clear?